Welcome to this scientific lecture on hydrogel microparticles and microcapsules. Today we're going to explore the fascinating properties and diverse applications of these remarkable materials that are revolutionizing fields from medicine to environmental science. Before we dive into microparticles and microcapsules, let's first understand what hydrogels are at their core. Hydrogels are three-dimensional cross-linked polymer networks that can absorb and retain large amounts of water while maintaining their structural integrity. These materials have the remarkable ability to absorb anywhere from 10 to 1,000 times their dry weight in water. This is due to the presence of hydrophilic functional groups in the polymer chains. Here's a visual representation. On the left we see the dry polymer network with crosslinks represented as white nodes. On the right, when water molecules shown in green penetrate the network, the hydrogel swells significantly while maintaining its structure. Now let's discuss the critical difference between microparticles and microcapsules. Both exist at the micrometer scale, but their internal structures are fundamentally different. Hydrogel microparticles have a solid matrix structure throughout their entire volume. The polymer network extends uniformly from the surface to the core. Microcapsules, in contrast, have a hollow core surrounded by a hydrogel shell. This shell core architecture allows them to encapsulate cargo within the internal cavity. Here we can clearly see the difference. The microparticle on the left shows a uniform distribution of polymer chains throughout. The microcapsule on the right has a distinct shell structure in white with a hollow core containing encapsulated cargo shown in yellow. Both microparticles and microcapsules typically range in size from 1 to 1,000 micrometers in diameter. This size scale makes them ideal for many biological and pharmaceutical applications. Let's now examine the key physical and chemical properties that make hydrogel microparticles and microcapsules so valuable in scientific applications. The first critical property is biocompatibility. This is essential for any material intended for medical or biological use. The high water content of hydrogels closely mimics the aqueous environment of natural biological tissues. This similarity reduces immune responses and allows cells to interact favorably with the material. Many hydrogel formulations are non-toxic and biodegradable, breaking down into harmless products that can be naturally eliminated by the body. Materials like alginate, chitosan, and certain synthetic polymers offer excellent biocompatibility. The second remarkable property is stimuli-responsive behavior. This is one of the most exciting aspects of modern hydrogel design. Hydrogels can be engineered to respond to various environmental stimuli, including pH, temperature, light exposure, and ionic strength. These triggers cause reversible changes in swelling or structure. Here's an example of pH-responsive behavior. At low pH on the left, the hydrogel microparticle is in a collapsed state with minimal water content. As pH increases, ionizable groups become charged, creating electrostatic repulsion that causes the particle to swell dramatically. A classic example is poly-N-isopropylacrylamide, or PNIPAM, which has a lower critical solution temperature around 32 degrees Celsius. Below this temperature, it's swollen, above it, collapses. This sharp transition near body temperature makes it ideal for drug delivery. The third important property is controlled permeability, which determines what molecules can enter or exit the hydrogel structure. The mesh size of the polymer network controls which molecules can diffuse through. Small molecules pass freely, while large proteins or cells may be excluded. This creates a selective barrier. The mesh size Z can be estimated from the molecular weight between crosslinks MC, the polymer concentration, and structural parameters. Typical mesh sizes range from 1 to 100 nanometers. The fourth property we must consider is the mechanical characteristics of these materials. Hydrogel microparticles exhibit a wide range of mechanical properties. Young's modulus can vary from 0.1 to 1,000 kilopascals, depending on cross-link density and polymer concentration. This tunability is crucial because we can match the mechanical properties of specific tissues. For example, brain tissue is very soft at around 1 kilopascal, while cartilage is stiffer at several hundred kilopascals. Let's pause here and address an important question. Why is cross-linking so critical to hydrogel properties? This is fundamental to understanding how we design these materials. Without crosslinks, polymer chains would simply dissolve in water and disperse. The crosslinks create permanent or semi-permanent connections between chains, forming an insoluble three-dimensional network. Higher crosslink density creates a tighter network with smaller mesh size. This results in stiffer mechanical properties and reduced swelling capacity. It's a trade-off we must optimize for each application. Conversely, lower crosslink density allows greater swelling and creates a softer, more deformable material with larger mesh pores that permit easier molecular transport. Now let's discuss how we actually fabricate these hydrogel microparticles and microcapsules. There are several sophisticated techniques employed in research and industry. The first common method is emulsification. This is one of the most versatile and widely used techniques. In emulsification, we create either water in oil or oil in water emulsions. The aqueous phase contains the polymer precursors, while the oil phase acts as a continuous medium to disperse and stabilize the droplets. Droplet size is controlled by adjusting stirring speed, surfactant concentration, and viscosity ratios. Higher stirring speeds and better surfactants produce smaller, more uniform droplets. Once the emulsion is stable, we initiate polymerization or cross-linking reactions within the droplets. This solidifies them into discrete microparticles. The continuous phase is then removed by washing. The second method is microfluidics, which offers exceptional control over particle properties. Microfluidic devices use channels with dimensions of tens to hundreds of micrometers. By precisely controlling flow rates of different phases, we can generate highly monodispersed particles with coefficients of variation below 5%. Here's a schematic of a microfluidic device. The polymer solution in blue and oil phase in orange meet at a junction. Due to the precise flow control, uniform droplets are generated at regular intervals, creating perfectly sized microparticles. Microfluidics also enables the fabrication of complex architectures. We can create core shell structures, multiple layers, or even Janus particles with two distinct hemispheres by using multi-phase flow designs. The third method is spray drying, which is particularly useful for large-scale production. In spray drying, a polymer solution is atomized into fine droplets using a nozzle. These droplets are sprayed into a hot chamber, where rapid solvent evaporation occurs, solidifying the particles. The high temperature causes almost instantaneous solvent removal. The dried particles are then collected from the chamber. This method is scalable and continuous, making it attractive for industrial applications. The fourth method is ionic gelation, particularly useful for natural polymers like alginate. In this method, droplets of alginate solution are formed and then exposed to a solution containing calcium ions. The calcium ions cross-link the alginate chains instantaneously, forming solid beads. The mechanism involves the negatively charged carboxylate groups on alginate chains binding to positively charged calcium ions. This ionic interaction creates the cross-linked network structure. Ionic gelation is extremely gentle because it occurs at room temperature without harsh chemicals or UV radiation. This makes it ideal for encapsulating living cells, proteins, or other sensitive biological materials. Now let's explore the diverse applications of hydrogel microparticles and microcapsules. We'll start with biomedical applications, which represent one of the most active areas of research. Drug delivery is perhaps the most prominent application. Hydrogel microparticles offer tremendous advantages over conventional drug formulations. Encapsulation within hydrogel microparticles protects sensitive drugs from enzymatic degradation, pH changes, and other harsh conditions in the body. This is crucial for protein and peptide drugs that are normally unstable. The polymer network controls drug diffusion rates, enabling sustained release over hours, days, or even months. This reduces dosing frequency and maintains therapeutic drug concentrations with minimal fluctuations. This comparison illustrates the difference. Conventional dosing on the left shows rapid concentration spikes followed by drops, requiring frequent administration. Hydrogel delivery on the right maintains steady therapeutic levels through controlled release. By functionalizing the surface with targeting ligands like antibodies or peptides, we can achieve site-specific delivery. The particles accumulate preferentially at the target tissue, reducing systemic side effects. The second major biomedical application is cell encapsulation and tissue engineering. This is a rapidly growing field with enormous therapeutic potential. Living cells can be encapsulated within hydrogel microcapsules where they remain viable and functional. The hydrogel provides mechanical protection while allowing nutrient and waste exchange through the permeable shell. A prominent example is the encapsulation of insulin-producing pancreatic islet cells for diabetes treatment. The microcapsules protect the cells from immune attack while allowing insulin release in response to glucose. Hydrogel microparticles can serve as injectable scaffolds for tissue regeneration. They can be loaded with cells and growth factors, then injected minimally invasively to promote healing of damaged tissues like cartilage or cardiac muscle. The third biomedical application is in biosensors and diagnostic devices. The stimuli-responsive properties are particularly valuable here. Hydrogel microparticles can be designed to change color or fluorescence in response to specific biomarkers like glucose, pH, or disease-related proteins. This provides a visual readout of the analyte concentration. Glucose-responsive hydrogels containing phenylboronic acid groups bind glucose and undergo conformational changes. This can trigger optical changes or controlled insulin release, enabling both sensing and therapy. Beyond biomedicine, hydrogel microparticles have important environmental applications. Let's examine some key examples. Water purification is a critical environmental challenge where hydrogels show great promise. Hydrogel microparticles can be functionalized with groups that bind heavy metals like lead, cadmium, or mercury. They also effectively absorb organic dyes and other pollutants from contaminated water through electrostatic or hydrophobic interactions. The microscale size provides a high surface area to volume ratio, maximizing contact with pollutants. This enables rapid adsorption kinetics and high removal efficiency, even at low adsorbent concentrations. Many hydrogel adsorbents can be regenerated by changing pH or ionic strength to release the captured pollutants. This allows multiple use cycles, making the process more economical and sustainable. The fifth application area is agriculture, where hydrogels can significantly improve crop productivity. Hydrogel microparticles mixed into soil act as water reservoirs. During irrigation or rainfall, they absorb excess water. During dry periods, they slowly release this stored water to plant roots, reducing irrigation needs. Fertilizer nutrients can be encapsulated within hydrogel particles. This provides slow, controlled nutrient release over the growing season, reducing losses from leaching and improving nutrient use efficiency. These applications are especially valuable in water-scarce, arid, and semi-arid regions where efficient water use is critical for agriculture. Field studies have shown irrigation water savings of 20 to 40%. Hydrogel microparticles and microcapsules also have fascinating applications in food science and technology. Encapsulation technology is widely used in the food industry to protect and control the release of flavors and nutrients. Many vitamins, omega-3 fatty acids, and flavor compounds are sensitive to oxygen, light, and heat. Encapsulation in hydrogel microcapsules provides a protective barrier that extends shelf life. Some nutritional supplements have bitter or unpleasant tastes. Microencapsulation can mask these off flavors while allowing controlled release in the digestive system where taste is not perceived. pH-responsive hydrogels can be designed to remain intact in the acidic stomach environment but dissolve in the neutral small intestine. This enables targeted delivery of probiotics or nutrients to specific locations in the digestive tract. Another food application is using hydrogel microparticles to modify texture and replace fat content. Hydrogel microparticles can mimic the mouthfeel and creamy texture of fat droplets in food products. This allows the formulation of reduced fat versions of products like mayonnaise, dressings, and dairy products. Since hydrogels are primarily water, they provide texture and volume with minimal calories. This supports the development of healthier food products that maintain consumer-acceptable sensory properties. Now let's address an important question. What are the current limitations and challenges facing hydrogel microparticle technology? Understanding these is crucial for advancing the field. The first challenge is mechanical weakness. Highly swollen hydrogels are soft and can be easily damaged by mechanical stress. This limits applications requiring robust mechanical properties. Researchers are addressing this through innovative designs like double network hydrogels, which combine two interpenetrating networks with different properties. Nanocomposite hydrogels, incorporating nanoparticles or nanofibers, also show enhanced mechanical strength. The second challenge is scaling up production. Many fabrication methods like microfluidics work well in the laboratory, but are difficult to scale to industrial quantities. Solutions include parallelized microfluidic devices that run many channels simultaneously, and optimization of continuous processes like spray drying for more consistent quality at larger scales. The third challenge is long-term stability. Some hydrogels degrade or lose their properties over time, especially in biological environments with enzymes. This is being addressed through more stable cross-linking chemistries, protective surface coatings, and selection of inherently more stable polymer systems. The fourth challenge is regulatory approval for medical applications. New biomaterials face extensive safety and efficacy testing before clinical use. Strategies include using already FDA-approved materials in novel formulations and conducting comprehensive biocompatibility and toxicology studies to expedite regulatory pathways. Let's now look at some exciting recent advances and future directions in this rapidly evolving field. The first exciting advance is four-dimensional printing of hydrogels. This adds time as the fourth dimension. 4D printing involves 3D printing hydrogel structures that are programmed to change shape in response to environmental triggers like temperature or pH. This enables self-assembling or self-folding structures. Potential applications include soft robots that adapt their shape to navigate complex environments, and biomedical devices that self-deploy or conform to tissue geometry after minimally invasive delivery. The second advance is the development of smart theranostic systems that combine therapy and diagnostics. These systems can sense disease markers and respond by releasing therapeutic agents in a feedback-controlled manner. For example, a particle that detects inflammation and releases anti-inflammatory drugs proportional to the signal. This enables personalized medicine, where treatment is continuously adjusted based on real-time monitoring of the patient's condition, optimizing therapeutic outcomes while minimizing side effects. The third exciting direction is biomimetic and bio-inspired hydrogel designs that replicate natural structures and functions. Researchers are creating hydrogel microcapsules with membranes that mimic cell membrane properties, including selective permeability, receptor-mediated recognition, and even simplified metabolism. This leads towards synthetic biology applications like artificial cells and protocells that can perform programmed functions. These could serve as drug factories, biosensors, or models for studying fundamental biology. To help solidify these concepts, let's think about a helpful analogy. How is a hydrogel microcapsule similar to a water balloon? A water balloon, shown here, has a flexible rubber shell containing liquid water. Similarly, a hydrogel microcapsule has a flexible polymer shell containing an aqueous interior. Both can swell or shrink based on internal pressure. However, there are critical differences. The hydrogel shell is permeable to small molecules, allowing exchange with the environment. The shell itself can respond to stimuli and change properties, and the shell is biocompatible for use in living systems. Let's work through a quantitative example to understand drug release kinetics from hydrogel microparticles. Let's consider a spherical hydrogel microparticle with a diameter of 100 micrometers loaded with a drug at an initial concentration of 50 milligrams per milliliter. Drug release often follows first-order kinetics described by this equation. Mt is the cumulative amount released at time t, m infinity is the total amount, and k is the rate constant. If the rate constant k is 0.1 per hour, what time is required for 90% drug release? We set mt over m infinity equal to 0.9 and solve. 0.9 equals 1 minus e to the negative 0.1 t. Rearranging gives e to the negative 0.1 t equals 0.1. Taking the natural logarithm and solving for t gives approximately 23 hours for 90% release. This demonstrates how we can quantitatively predict and design release profiles. Let's summarize the key concepts from this lecture on hydrogel microparticles and microcapsules. First, hydrogels are three-dimensional cross-linked polymer networks capable of absorbing large amounts of water while maintaining structural integrity. Second, microparticles have a solid matrix structure throughout, while microcapsules have a shell core architecture with a hollow interior. Third, critical properties include biocompatibility, stimuli-responsive behavior, controlled permeability, and tunable mechanical properties. Fourth, various fabrication methods including emulsification, microfluidics, spray drying, and ionic gelation enable different particle structures and production scales. Fifth, Applications are remarkably diverse, spanning drug delivery, tissue engineering, diagnostics, water treatment, agriculture, and food technology. Sixth, current challenges include improving mechanical properties, scaling up production, ensuring long-term stability, and navigating regulatory pathways. Finally, exciting future directions include 4D printing, smart theranostic systems, and biomimetic designs inspired by nature. Let me close with a final question. What makes this field so exciting for researchers, and why should we be optimistic about the future? The field brings together chemistry, materials, science, biology, engineering, and medicine in a highly interdisciplinary way. This convergence accelerates innovation and enables solutions to complex problems. The technologies we've discussed have tremendous potential to improve human health through better medicines and diagnostics, and to protect our environment through more efficient resource use and pollution control. Our fundamental understanding of hydrogel behavior, especially at the micro and nanoscale, continues to evolve. New discoveries regularly open unexpected possibilities and applications. The tools available for designing and fabricating hydrogel microparticles are becoming increasingly sophisticated, from advanced microscopy to computational modeling to precision manufacturing. This enables ever more complex and functional designs. Thank you for joining me in this exploration of hydrogel microparticles and microcapsules. I hope this lecture has given you a comprehensive understanding of the fundamental properties, fabrication methods, diverse applications, and future potential of these remarkable materials. The field continues to advance rapidly, and I encourage you to follow new developments in this exciting area of science and technology.